Hi there, this is Mark Cannon. I'm going to have a few minutes to discuss with you our study on the mitochondrial and transcriptome study of autistic and non-Western diet children. First of all, there is an association between the gut microbiota and autism spectrum disorder. This was published in Frontiers in Psychiatry, a review and meta-analysis of 9 studies. And by the way, this was one just done last year, but there has been others previously. In addition, in 2017, published in Scientific Reports, the study Alterations of Oral Microbiota Distinguished Children with Autism Spectrum Disorders from Healthy Controls. We absolutely, after you hear this study, we would concur with that 100%. So this study, we looked at 30 children with autism spectrum disorder, very well diagnosed, severe autistic spectrum disorder children from my private practice, and compared them to 30 children from the country of Colombia that were healthy and neurotypical children. So we took sampling at 3 times over 60-day periods from the kids in my private practice, along with mitochondrial swabs. And so we had a full range of the full transcriptomics, which is the entire viral, bacterial, archaeal, and fungus range of species, along with all the functional groups of the KEGG orthology. And we did the exact same for the Columbia component, except for it was a one-time sampling. And we got their mitochondrial cells and also from buccal cell swabs and their mitochondrial health. And then we also did the transcriptome salivary sampling. There was very significant differences between each group, and it was very shocking, the completely different number of unique taxas in each group, and then compared to the healthy controls to those with kids with autism, and then those with the kids with autism after being exposed to 60 days of xylitol, and again after a 60-day exposure to probiotics. And then we also had the mitochondrial results, except for those from Colombia got heat degraded. So the KEGG orthology was a wealth of information, unbelievably, because we had all the functional groups and the RNA transcripts that were for all the enzymes and all the peptides and all the chemical messengers and all the neurotransmitters. He found some very, very unique things that have been going on in the kids with autism affecting their neural development. The statistics were quite amazing, as you can see from the poster. Our Spearman correlations, we had some very tight grouping of the different groups, and it was fascinating, the difference between those kids that responded very strongly to xylitol, because 6 of the kids that were extremely nonverbal became extremely verbal and had tremendous changes in their capabilities after 60 days of xylitol treatment. The xylitol was simply to see the shift in the oral microbiome, which we expected to see, but we did not expect to see such a big neurological improvement. Here's on the mitochondrial health. Again, we had some significant differences between the groups. So basically, the transcriptome of individuals with autism, it's quite a bit different from those of healthy children in a developing country. And in fact, we've actually had another subgroup that we've done of this healthy children in the United States. And again, another big difference there. But we also note that these microbiome changes may have very, very strong epigenetic consequences. Thank you for listening.